Hello everyone, welcome back. So today we are going to see about the host microbial interactions. Okay, a very important topic. Okay, host microbial interactions. So host microbial interaction will play a very significant role because whatever future aspects I'm telling, it is going to depend upon the host microbial interaction. So if I say that there is going to be destruction of collagen, there's going to be bone loss, yes, it is linked to this video. So be very attentive and be very careful while watching this video, because once your base is strong, you can build over it, you can build n number of facts over it. Okay, so the host microbial interactions, it is just to tell you that what happens when a bacteria enters the body and how a body responds to it, and what does the bacteria do in return, and what happens to the tissues. Host is us, microbes is the bacteria, we do interact. What is the response of a body? Okay, very simple. So any bacteria, say for example Porphyromonas gingivalis or Aggregatibacter actinomycetum comitans, any bacteria for that matter, okay, it will have certain patterns over itself. These patterns are extremely unique for each bacteria. Okay, so these patterns are called as the pathogen pathogen associated, okay, pathogen associated molecular pattern. Okay, so this pattern which is formed by each bacteria is extremely unique. It is called as pathogen associated molecular pattern, or PAMP. Okay, so whenever a bacteria tries to enter the body, it has to enter the epithelium, and then only it can penetrate into the connective tissue. Always remember, the epithelium of the gingiva, especially your circular and junctional epithelium, is non-keratinized. It is semi-permeable because it lacks the waterproof layer. Okay, so the PAMP which is present in the bacteria, it is going to come in contact with the epithelium, isn't it? So it comes in contact with the epithelium. Say for example, in this case, it is junctional epithelium. Okay, so the bacteria which is present in your block or a calculus enters the sulcus, comes in contact with the junction epithelium. Each bacteria will have a PAMP. So what our body will do is, there will be cells of innate immunity. Do you have any different cells in your epithelium? Obviously you will have. We will have cells of innate immunity in the epithelium. Say for example, natural killer cells, dendritic cells, mast cells, tissue macrophages, all these will be present in the epithelium. All these will have the cells of innate immunity. What is the first line of barrier? Epithelium. Which is the first line of defense? Neutrophils. First line of barrier is different, first line of defense is different. Okay, now this bacteria which has PAMP enters or tries to enter the epithelium. So what our body will do? Our body will see, okay, some enemy is trying to enter the territory. We'll call our friends and we will fight and push him outside. That is a normal tendency of any cell when a foreign object enters, right? So similarly, to recognize that PAMP, our body will have certain receptors called as pattern recognition receptors. Pattern recognition, which what pattern it recognizes? This pattern. This pattern it recognizes. So I'm going to call that as PRR, pattern recognition receptor. Okay, these pattern recognition receptors are nothing but toll like receptors. What are they? Toll like receptor. Okay, say for example in this case a NK cell, natural killer cell, will have toll like receptor, which is nothing but pattern recognition receptor. What pattern it recognizes? The PAP, the pathogen associated molecular pattern. So this toll like receptor goes and engages into the PAP. So once it identifies the PAP, which is nothing but a foreign object, what it will do is it will start to synthesize what is called as pro-inflammatory cytokines. What it is called as pro-inflammatory cytokines. So pro-inflammatory cytokines, can you name some pro-inflammatory cytokines? Exactly. Interleukin 1, interleukin 6, interleukin 8, tumor necrosis factor alpha. All these are pro-inflammatory cytokines. How it is stimulated? By NK cells. Cytokines are nothing but proteins. Okay, pro-inflammatory, before inflammation. So a body sends signals so that the inflammatory cells comes out. Okay, so what does pro-inflammatory cytokines do is it does a lot of job. One thing what it does is it is going to stimulate the endothelium. It is going to stimulate the endothelium so that the cells of inflammation comes out. Okay, exactly. So the first step in inflammation is there is going to be transient vasoconstriction. The endothelium is going to compress or is going to constrict for a shorter period of time, very short period of time, which is followed by persistent vasodilation. So imagine you're having a small room. Okay, a small room will have some 3 to 4 feet of opening. Okay, I'm asking 100 people to come out of that room at the same time. Will it be difficult or easy? Definitely it will be difficult. You'll squeeze through each other and you'll come out. Now in the same room I'm asking 2 people to come out. It will be very easy, right? Similarly, when the endothelium constricts, it will be extremely pressure inside the endothelium. Similarly, when endothelium dilates, what will happen? The cells are freely moving now. They are very slowly moving. So what happens? There is going to be increase in viscosity. So because of the increase in the viscosity, what is going to happen is the neutrophils, which is the first line of defense, it is going to go and attach, or it is going to move towards the endothelium. Okay, there's going to be margination. Okay, so because of the stasis is nothing but the slowing down of the blood vessels or the blood cells. Okay, so because of the stasis there is going to be the attachment of the neutrophils to the walls of the endothelium. Okay, in the walls you will have a lot of receptors. Okay, say for example P selectins, E selectins. Okay, and then you will have ICAM, intracellular adhesion molecules, VCAM. All these are adhesion molecules. Okay, so what will happen is these neutrophils, they go and adhere to the endothelium. Okay, once they start to roll over the endothelium, it will come in contact with these integrins, selectins, and ICAM, VCAM. So what happens is the P and the E selectins, as the neutrophils roll over the P and the E selectins, there is going to be temporary adhesion. There is going to be temporary adhesion, which means the neutrophils is not firmly attached to the endothelium, but it makes sure that I'm coming. Okay, so what happens? The P and the E selectins, they are going to give handshake to the neutrophils and send them off. And then as the neutrophils rolls over, it comes in contact with ICAM and VCAM, that results in permanent adhesion. that results in permanent addition. So once the neutrophils, okay, once the neutrophils they starts to roll, comes in contact with the P and the E selectins and ICAM and VCAM, they become firmly attached to the walls of the endothelium. Okay, so once these are firmly attached to the endothelium, what will happen is, just I'll try it here. Once these are firmly attached to the endothelium, they start to move out of the endothelium by a process called diapedesis. Okay, so with the help of diapedesis, the cells of the inflammation, they move outside, and they, because the cells are widely spaced, there's going to be increased vascular permeability, because it is one of the feature of inflammation, where there will be increased vascular permeability, increased hydrostatic pressure, vasodilation, all these will take place. Because of the increased vascular permeability and hydrostatic pressure, there will be formation of edema, and also it will help neutrophils to come out of the endothelium by the process called diapedesis. And once the neutrophils comes out, now we have to reach the site or reach the bacteria to destroy it, right? So these neutrophils, okay, they have to reach the bacteria which is present here. That is when it is going to destroy the bacteria, isn't it? Fine. Now the process of migration of a neutrophils to the site of bacteria is called as chemotaxis. Who does this job? Chemotaxis is a migration of neutrophils to the site of bacteria. Who does this job? Can anyone tell me? Exactly. So, as I told you, there are a few pro-inflammatory cytokines secreted by toll-like receptors, which is nothing but pattern recognition receptors. So, these toll-like receptors are otherwise called as chemokine. They are otherwise called as chemokine. So, the function of interleukin 8 is, once the neutrophils comes out by a process called diabetes, it is going to give signals to the neutrophils, come, I'll take you to the enemy, like it is a vehicle. Okay, so the neutrophil sit over, or it will receive the information by the interleukin 8, which is a chemokine, and it will slowly move towards the site of bacteria. And once it reaches the bacteria, it is going to destroy or kill the bacteria by the process called phagocytosis. Okay, so the phagocytosis can be taken by oxygen dependent or oxygen independent. Okay, so either they can release reactive oxygen species into the bacteria and destroy them, or they release some enzyme which will destroy the bacteria. So in this way, a neutrophil kills the bacteria. And what happens after killing the bacteria? So after the bacteria is killed, these neutrophils, they are going to present these bacteria in the form of antigen via MHC2 complex. Via MHC2 complex, major histocompatibility complex, HLA. Okay, so via these complexes, the neutrophils, after engulfing the bacteria, after destroying the bacteria, it is going to present these bacteria as antigens via the MHC2 complex. What is the function of this MHC2 complex? Very simple. This MHC2 complex, it is going to stimulate the T lymphocyte. It is going to stimulate the T lymphocyte. So what is the T lymphocyte is going to do? It is going to stimulate B lymphocyte, and that is going to stimulate plasma and memory cells. Plasma cells produce antibodies, and memory cells make sure that the next time if it comes, I'm going to attack it very easily. Okay, this is how the innate and the adaptive immunity works. Okay, so meanwhile this interleukin 1, 6, 8, TNF alpha, apart from stimulating inflammation, apart from doing a very good job, it also does a few bad things also. Okay, say for example, this interleukin 1, 6, tumor necrosis factor alpha, all these are called as bone resorbing factors. Okay, all these are going to cause bone resorbing factors. How? Because it is going to increase the expression of rankle, rank and rankle. All these are receptors which is present in your rank is present in osteoclast and rankle is present in osteoblast. So once these receptors are expressed, bone resorption takes place. And also these pro-inflammatory cytokines is going to stimulate prostaglandins, which is going to increase or stimulate bone resorption. And these pro-inflammatory cytokines also stimulate MMPs. What is MMP? Matrix metalloproteinases, which will release collagenases, which will destroy the collagen that results in pocket formation. Okay, so apart from good things, these pro-inflammatory cytokines, they also cause bone resorption, they also stimulate collagenases enzyme pocket formation. That is the reason why when the bacteria enters into the periodontium, even though the body tries to kill the bacteria, there is bone resorption because of all these factors. Okay, so now am I right in telling that any systemic problem, say for example diabetes, if a patient is having diabetes, there will be definitely increased amount or increased synthesis of pro-inflammatory cytokines, so obviously there will be bone resorption. That is how a link between diabetes or any systemic disease to perio, whenever a systemic disease is having increased pro-inflammatory cytokines. So there is pro-inflammatory cytokine increase in systemic problems. There will be definitely periodontitis. There will be definitely a link between periodontitis and pro-inflammatory cytokines increased diseases like diabetes, cardiovascular diseases. Okay, so in all these situations, it is just because of the pro-inflammatory cytokines. Fine. So this is how the normal bone resorption or a pocket takes place. Okay, now what will happen to the neutrophils which have already migrated to the site? So what will happen to these neutrophils? So these neutrophils, what will happen to these neutrophils? Where thousands and thousands of neutrophils have migrated. Yes. Then what will happen is, instead of synthesizing pro-inflammatory cytokines, once the inflammatory process has ended, that is the bacteria is destroyed, our body tries and produces and synthesis what is called as the anti-inflammatory mediators. So anti-inflammatory mediators are interleukin 4, interleukin 10. What it will do? It is going to result in destruction of neutrophils. Okay, it is going to result in destruction of neutrophils by apoptosis. I'll tell you a few mediators of resolution of inflammation. See, when the inflammation is ended, it results in resolution of inflammation. Resolution, that is, I'm going to revert the tissues to normal. Resolution of inflammation. So I'll tell you a few mediators of resolution of inflammation. Okay, lipoxins, resolvins, maricins, protectins. Lipoxins, resolvins, maricins, protectins, all these are mediators of resolution of inflammation. Okay, so when a body responds to an infection, responds to a microorganism, we call that as inflammation. Once the inflammation is over, our body again send signals that okay, everything is done. You have to get rid of the remaining neutrophils or neutrophils which are migrated to the area. It sends lipid mediators of inflammation, that is lipoxins, resolves marasins, protections, and stimulates anti-inflammatory cytokines. So the process of inflammation retards there. Okay, so this is how broadly what happens when a bacteria enters the body. PAMP, PRR, rank, rankle. You just get familiarized with the terms. See, rank, rankle, I will definitely speak about when I talk about bone resorption. Just know what or how this rank and rankle are produced. It is produced by pro-inflammatory cytokines as well as prostaglandins. Then what is pro-inflammatory cytokines? You should not blink. Now pro-inflammatory cytokines synthesized by toll-like receptors, which is nothing but pattern recognition receptors. So this is a cycle. This will be the very basic overview. So if you're not understanding, you can just play it again, but make sure you are completely thorough in this video and you should have zero doubts. So once you have zero doubts in this video, whatever future aspects I'll add on to it, you can easily understand. Okay, so overview of how a body responds to a bacteria is seen. Now when I tell you a bacteria enters the body with the virulence factor lipopolysaccharides, you will be able to correlate. Okay, fine, the bacteria will have PAMP, it recognizes, and at the epithelium it is recognized by PRR, so that is why it enters the body. Okay, so thank you so much for watching this video and have a great day. Thank you.